Hi everyone, so this is our final lab presentation. We're group number 8. I'm Lena.
I'm Stephanie.
And I'm Lennox. So to start off, in biology, environmental samples often contain several different organisms, so they have contaminations. And oftentimes what happens is that in experimentation, organisms have to be isolated to perform a lot of specific tests. It's really important to achieve this isolation before you start to identify your unknown sample to avoid having contamination and receiving incorrect results. Identification of an organism is really important in the lab setting, but it's also equally important in clinical settings where unknown organisms are identified to develop treatments based on what the organism is. In this experiment, several biochemical and differential tests were performed with the ultimate goal of identifying an unknown bacterial organism. So to start off, what we had to do was do differential stain to figure out what type of bacteria and its cellular morphology was. So we arrived at the conclusion that our bacterium was a gram-negative rod. From here, we followed the dichotomous key and started performing biochemical tests to determine what our unknown was, right? So after getting a negative oxidase test, we followed the family Enterobacteriaceae table, and we got to the conclusion by performing the biochemical steps. And there's the API 20E strep with our results on there as well. So here in week 4, we did a bunch of serial dilutions. At first, the concentration was a little bit too high, so we had to be able to serially dilute it to get a colony forming unit and a viable titer. Our CFU was 73 and our viable titer was 7.3 times 10 to the 8. Here on the left side, we see the colonial morphology was opaque. The colonies were small in size and the margins were entire. And finally, the elevation, it was raised a little bit. In week 5, we also see that the colonies had similar morphology to the week 4 ones. And when magnified under the microscope at 100 times, we see that this is where we came to the conclusion that we had gram-negative rods as well. And lastly, here on the right, this picture shows uh, like if our expectations were matched with our unknown sample, and spoilers, but yes, it was.
In week six, we did our biochemical tests. In figure 1, we had our OF medium. In both of the test tubes with oil and without oil, we had positive reactions and the pH was acidic in both. In figure 2, we had our Kligler's iron agar deep slant, where we had our test for the slant for the butt both to be positive, which meant that it was able to ferment all 3 sugars, sucrose, glucose, and lactose. In figure 3, we had our nitrate reduction broth, and our tube on the left with NO3 and the tube on the right without NO3. The tube on the left was positive for nitrate reductase, and there was no pH, and the tube on the right was negative for nitrate reductase. In figure 4, that was a negative test that we had for the nutrient gelatin slant. In figure 5, we did our starch hydrolysis, where our test was positive. And for figure 6, our catalase test was found to be positive, which indicated that we had the bubbles, and our oxidase test was negative because there was no color formation on the slide. And for figure 7, we had a variable plate for the urea agar. And over to the right, we have our week 7, where only 2 of our tests tested positive, which was the iosine methylene blue agar plate and the McConkey agar plate. And the MSA plate, as well as the blood agar plate, were both found to be negative. And the EMB plate, there was gram-negative lactose fermenting bacteria on that plate. And on figure 11, on McConkey agar, the pH was acidic and there was gram-negative bacteria on the plate. For week 7, we came with the question which enrichment media would best fit our bacteria, and we came up with Hectoin enteric agar. The reason we found this to be the best for our bacteria is because this agar plate works best with gram-negative bacillus bacteria, and that is what our identification of our bacteria was. In week 8, we did our API 20E strep test. The tests that were positive were ONPG, which tested the enzyme for the substrate nitrophenol B-D glucoperoxide. And we had the IND test, which tested positive for indinol, as well as the glucose test, which tested positive for glucose and that it was fermented, as well as MAN test, which tested positive for mannose, as well as the SOR, RHA, MEL, and ARA test, which tested for the arabinose. In figure 2, we were able to identify the code, and our code was 1044552. On the right, in week 9, we had our birdies identification. The first step was to identify which type of bacteria we had, which is gram-negative rods. And step 2, we went with the oxidase test, which was negative, which led us to step 3, which was a lactose test, which was positive. Step 4 was to do the indole test, which was positive. And step 5 was to conduct the citrate test, which was negative as well, leading to our identification of E. coli strain C. Week 10, we were able to identify the antibiotic resistance. We were able to see that tetracycline with our bacteria was susceptible, as well as carbonicillin and streptomycin. And we were able to find out the antibiotics that were resistant, which were erythromycin, penicillin, and bacitracin.
Okay, so going off our biochemical test results, we had 2 unexpected results that we obtained. So for the urease test in week 6, we got a variable plate because it showed both pink and yellow, so both positive and negative. But then in week 8, when we did the API 20E test, we got that it was just yellow, so it was just a negative result. And then that would just mean that our bacteria cannot produce and utilize the urease enzyme, actually. And then another unexpected result was the starch hydrolysis test. We originally got that it was a positive test, but then later on through research, we figured out that in fact, E. coli should give us a negative result. So that was another unexpected result. Okay, so in addition to the hectin enteric agar, enteric agar that Stephanie mentioned earlier, another good isolation method would be the tissue culture plate, because this can detect biofilm. So it can detect if our bacteria would be producing biofilm, and it can detect if it's going to produce it in high, moderate, or like not at all. And this test is going to use crystal violet staining and a 96 well plate. And there's actually an image used from it's actually an image gotten from an actual study done on our specific strain of E. coli, strain C, and it shows what kind of biofilm ours can produce. And then for the environment that our bacteria can be found in, so E. coli can be found in many different, it can be found in humans and animal intestines, as well as in agriculture, because since it's found in the intestine of animals, the bacteria is going to go out in the feces of the animal, and it's going to end up in water sources. It can end up in crops, the soil, and that causes contamination. The usefulness of studying this bacterium is going to be found in biopharmaceutical industry. These bacteria are used to study proteins and gene therapy with gene therapy and DNA vaccinations. So some harmless strains are often used in laboratory settings for multicellular bacterial research to study genetic compositions and gene expression, as we can see in the right image. This is actually the chromosome map of the E. coli strain C. This was taken from the same study that was studying the biofilm production.
To kind of wrap it all up, as we previously mentioned, E. coli is found in a lot of animal intestines. So after they do their business, it can mean profit loss for agricultural companies, and then it trickles all the way down to causing a loss of jobs in the agricultural industry, and it could potentially cause an economic depression and decrease in food supply. So in conclusion, identification of unknown bacteria is beneficial for both the health of the people, but also like to the economy. So by identifying the bacteria, we could combat it quickly and efficiently because we know what type of bacteria it is, right? And that's our presentation. Thank you guys for listening.